Welcome to Pharmacology lectures. Today we are going to start Pharmacodynamics. Pharmacodynamics is the action of a drug on the body. Now, what the drug does to our body. We have studied in kinetics what our body does to a drug. Now here it's vice versa. We have receptors on our cell membranes. Receptor and drug molecule, the binding of these two is just as a lock and key mechanism. Receptor with bound agonist is activated. It has first our cell membrane has receptors which are unoccupied. Once the drug arrives, it enters, it is accepted on the receptor, it binds towards it, then its physical and chemical properties are altered, which leads to cellular molecules to cause a biological response. Once the receptor is activated, there will be a signal transduction and you will receive a biological response, which we say drug effects. Now, signal transduction. Drugs act as signals and receptors act as signal detectors. They will detect the signals. Now, the term agonist is used when the protein which used to be bind on the receptors are same as the drug molecule, which will perform the same mechanism. It is known as agonist. It will copy the natural processes. The agonist will bind to the site of the receptor protein. It will activate into a series of reactions and it will result into a response. Second messenger or effector molecules are a part of cascade of events. Now, the drug and receptor complex. Cells have many different types of receptors. There is drug receptor complex. Now, cardiac cell membranes, for example, they have beta adrenergic receptors. We will also study in the CVS chapters. These receptors will bind and respond to epinephrine or norepinephrine. Okay, cardiac cells also contain muscarinic receptors, which are bind to acetylcholine. These two receptors will control the heart's vital functions. Now, the magnitude is dependent on the drug receptor complexes. This concept is also similar to the formation of coupling between enzyme and substrate we used to study in just an example of lock and key mechanisms. Now, there is an example of antacids, such as chemically they neutralizes excess gastric acid. Receptor states, either it will be inactive or it will be active, and they have reversible equilibrium. They can, if they are inactivated, they'll become active. We'll just go through it fast and we'll see the main things. Now, major receptor families. This is an important topic. The receptor is divided into 4: ligand gated ion channels, G protein coupled receptors, enzyme linked receptors, intracellular receptors. Now we will have a look here. Ligand gated ion channels are changes in the membrane potential, ionic concentration, that is, within the cell. Examples are cholinergic nicotinic receptors. G protein coupled receptors are alpha and beta adrenoceptors. Protein phosphorylation is occurring here. Alpha, beta and gamma. G coupled proteins. Now, the third is ligand binds to the extracellular domain of a receptor and it activates the kinase enzymes. Okay, enzyme linked receptors. Intracellular receptors, steroid receptors. Lipid soluble ligand diffuses across the membrane to interact with the intracellular receptor. In the A, ligand is binding to the extracellular domain for ligand gated channel. In B, ligand binds to a domain of a transmembrane receptor here. In C, ligand binds to the extracellular domain, and D, ligand diffuses across the membrane. Just this will not go into the deep. We can see here. Now, The first one, this is the extracellular space, this is cytosol, these are G coupled protein, they're bound with GDP. Hormone or neurotransmitter, a chemical arrives here, ligand arrives here, receptor it will receive. This is the cell membrane, this is inactive adenyl cyclase. Once the receptor is occupied, GTP converts into GDP. Next, what happens? It is transferred here, and then CMP converts into PPI. When the hormone is no longer present, the receptor reverts to its resting place. This is reversible process. It gets deactivated. Okay, we have seen this. Now this is an important insulin receptor inactive. First, it is inactive. This is tyrosine. Once the insulin arrives at its receptor site, it will bind and it will activate the receptor tyrosine kinase activity. This is an enzyme. It is being activated in the intracellular domain of the subunit for insulin receptor. The tyrosine residues of the beta subunit are autophosphorylated. Then receptor tyrosine kinase phosphorylates other proteins, for example insulin receptors, substrates, and thus it activates signaling pathways, and then we can see phosphorylation and biological effects of insulin. Okay, now you can see here, it's a big. Now what happens? This is a target cell, a lipid soluble drug. Okay, this is a lipid soluble drug. It will diffuse across the cell membrane, move into the nucleus. This is the receptor which is in its inactive form. Once it binds, it will become activated. It goes into the nucleus and it brings a chain of reactions, and then you can see the effect.